it is too small, it mix some neurons close by. If it is too large, we will get too much neurons, then it will be impossible, and this is also related with some insertion problems. But I would like also to remind that this is, for instance, for one textbook, say, when we are talking about the brain, usually we are talking about neurons, and say the major constituent of the nervous system are neurons, but this is not true, because you can see that most of the cells in the nervous system are not neurons. Glial cells, for instance, outnumber the neurons in one relation 10 to 1, but there is also blood vessels, there is also other kind of cells. So this is one slide from we take together with Pierre Ranshaw, Henri Divernois in our lab. The black things are blood vessels. This is high magnification. This is the surface of the brain. So it's full of blood vessels. It's almost impossible to avoid some blood vessels. And what happens when we try to put some electrode into the brain? Usually we got some microhemorrhage. And this is just one sample by using the UTAS electrode array. In this case, it's in human. You can see this is the array. When we remove the array, you can see the microhemorrhage on the surface of the brain. This is the histology. You can see here the needles where the electrodes are, but a higher magnification, there is many healthy neurons around. Other problems is not this was an acute experiment. What happened in the long term, in chronic experiments? We can find out some electrode trial like this with many healthy neurons around, but in this case you can see many dots, blue dots, that this is one chronic inflammation. So you see this is a normal electrode and this is one chronic inflammation. So in some cases there is some chronic inflammation that are damaging the neurons, are damaging the electrode, are damaging the electronic and so on. And there is also some encapsulation. The red color are glial cells. You can see some capsule is some encysted around the electrode. Then it's more difficult for us, for electrode, to get signal from the tissue, or if we try to send signals, if we try to send electrical stimuli, it's also more difficult. You can see all the gliosis around the electrode, and in any cases, we can even see something like this. This is a metal-like structure in the brain. It was by using some other kind of technology. When we remove the electrode, we can see this is a silicon electrode and this is platinum in this case. So we can make one microanalysis and we can see here, if we put the tip of the microanalysis here, we can see the silicon. In this case, there is also silicon, but DEX silicon. And what happened here, for instance, we see here the platinum, and here see less platinum. Here is close to 3, or 1, 2, something like that. And we see the silicon that is under the electrode. So the macrophagus, all the cells are eating the coverings of the electrode. So the electrode become damages. In order to avoid some of these problems, we are running the NeuroPros project, and the main objective of the project is to develop multifunctional microprobe arrays that able to communicate with the nervous system not only by the electrical signal, but also by chemical signal with fluidic, and with several important features like a lot of electrodes, dense three-dimensional, really three-dimensional microelectrodes with a modular technology, with microfluidics for delivering drugs, for delivering neurotransmitters, for getting even some fluid from the brain, with accurate deep control, and with several other features that we think they are going to be very important in the future. So the problem for us is to be able to record three-dimensional sheets of neurons in vivo and in long-time studies. So, and the system is able or is going to be able to work with electrical stimulation and even incorporate telemetry and so on. So these are different approaches. You can see the Michigan probes, mainly body electrodes, the UTAS approach, mainly tip electrodes, and this is our approach, the NeuroProbe approach, in which we have tip electrodes, but we also have body electrodes. And one of the key points in the project is the platform concept. So we have the system is able to deliver microfluidic to deliver electrical signal and so on. And this was the idea, but you can see here that we already have some of the platforms, and I will show you later some of the recording, and the system seems to work very, very nicely. So these are several prototypes of the system. So before, this was one of the first prototype, maybe this was one of the first prototype, even the connector, you can look that is changing. Also because the number of electrodes is changing also. Here we have, I don't remember exactly, 9 electrodes, and now we are working with system with 72, and even we have now one system with 500 something electrodes. So this is one of the last prototypes. In this case, it's 4x4, and this is the connector. And I can show you that we have system with 4 millimeters probes, with 8 millimeters probe. We have now something with 2 millimeters, and we can configure everything as tetrode and other different kind of approaches. So, and also we are developing, or we are working on the amplifiers. They are active probe. You can see here how it looks like. Of course, we also have integrating not only microfluidics, but also some of the partners of the project are working on biosensors, and we already have some biosensors for choline and for glutamate. You can see here, this is from Milena Kudelka from Neuchâtel, and the system is able to work nicely, and everything is integrated in the same needle. So the same device allows us to deliver drugs, to get work as a biosensor, to get signal as a, not only, action potential, we are trying to get also local field potentials, and so on. So we have a modular technology for these probe arrays that allow us some customization. It's scalable, we can reconfigure the system, incorporate microfluidics and biosensor, and of course electrical on the same platform. Is a slim profile because we are using the system in big animals, in cats and so on, even in monkeys. We need that it should be floating into the brain. And we are working not only, as I told you, with local field potentials, but also spikes in rats, monkeys, and even humans. I would like just to show some of the samples. In this case, it's in vivo experiments. We can see some of the needles. The noise level is very good, less than 25 microvolts, and we are able to get spikes with a spike as amplitude 50 to 150 microvolts. You see different neurons from the same electrode. We are also developing the software for getting or for the clustering or for knowing how many neurons are we recording from. And this is what happened this is a 2-month experiment. We see that, of course, there is some gliosis